Alrighty, everybody. Thank you for joining me today. As was mentioned earlier, my name is Susie and I'm a PhD student from Flinders University in Australia. And today I'm going to be sharing some of my work where I've been modelling bacteriophage protein complexes. So in case you don't know already, bacteriophages or phages are viruses that infect bacteria. And sometimes they look a little bit like spaceships, like you can see on the slide here. Bacteriophages are actually the most abundant and diverse entities on Earth. And because of this, they have a lot of really important roles in a range of different systems, both within human health and within the environment. Like any organism, phages are made up of proteins, but one thing that makes them really difficult to study is that about 65% of the proteins within bacteriophages have no known biological function. So during my PhD, I've been working on developing methods that we can use to predict the functions of unknown genes in bacteriophages. One way that I've been doing this is by using AlphaFold, and you can see here on this slide that using AlphaFold, I've been able to generate protein structures for a range of different phage proteins, things involved in their structures, so things like head and packaging genes and the genes for their tail, but also genes for things like DNA replication and other metabolic genes as well. While using these protein structures is really helpful for understanding phage proteins, they don't always necessarily tell the whole story. And the reason for that is because using AlphaFold, I can generate a protein structure. Like here, for example, I can take the major capsid protein, one of the proteins on the head of the phage, and generate a pretty good structure using AlphaFold. However, in reality, this protein actually doesn't just exist alone. And the different Lego pieces of proteins actually interact together to form bigger structures. So for example, this major capsid protein actually forms a hexameric protein with 6 different subunits. And we could say then that this protein has an oligomeric state of 6. And just by generating this AlphaFold structure of the single monomer, we wouldn't necessarily know this information and have the whole picture of what this protein looks like within the virus. So I've been working on a way where we can predict what this larger complex is that this protein might form. And the way that I've been using this is I've been using CollabFold, which is kind of a version of AlphaFold that's been made more accessible. Essentially what I do is I take a phage protein that I am interested in and build a multiple sequence alignment of it using the search within CollabFold and the databases that are present there. Once I've generated this alignment, I can then specify in the alignment how many subunits I would like in my folded structure. And then once I've done this, this model gets folded using AlphaFold with the number of subunits that I've specified. So for example, here it was 3, so I'm going to get a complex here with 3 subunits. I can then repeat this for a range of different subunit sizes. So for this major capsid protein here, I've done this from 2 through to 9 different subunits and generated structures for each of them. And you can see just visually here, right, that when we use too few subunits, for example, when we use just 4, the structure doesn't look quite complete. And when we use too many here, like 9, you can see it starts to look a little bit like spaghetti. So ideally, we wouldn't want to just visualize this. We would want to quantify this. So the way that I quantify this is by looking at the quality of the interfaces between the different subunits within the structures. So looking at the quality of the residues within those regions. And I can do this using the IPTM score. And what we can actually see here on this plot is that this IPTM score here peaks when 6 subunits are used. And this is consistent with what we know about the major capsid protein of this virus. So now that we can kind of see that this process of folding the protein for a range of different subunit sizes and looking at the interfaces could be useful for inferring its number of subunits in its complex, I wanted to test this further. So what I did was I went to the protein data bank and I was able to find about 2,000 phage protein structures. I went and filtered these down further, removing anything that was heteromeric because that's kind of out of the scope of this, and removed anything that was synthetic or mutated or anything like that. And I was left with 215 phage protein complexes to investigate further. With these 215 proteins, I then folded them using that method I described earlier, repeating it for 2 to 10 different subunit sizes for each one. And then once I had these here, I calculated the metrics, looking at the quality of the interfaces, and then evaluated what types of complexes each of these proteins might form. In doing this, the first thing that I wanted to look at was whether or not I could predict whether a particular protein was a monomer or a homomer. So here I had 37 monomers and 178 homomers. And what I did for each of these proteins was I looked at the quality of the interfaces that they form. And essentially what I found is if I have a monomeric protein and I'm forcing it into some bigger structure, unsurprisingly, the quality of its interfaces aren't so good. And when we have a look at these metrics here, we can see that the monomers have very poor interface metrics and that the homomers have better interface metrics when they're in their correct state. To see if this was predictable, I made a simple machine learning classifier, a logistic regression model, and by using some of these different quality metrics, I was able to attain 91% accuracy in predicting whether one of these phage proteins was a monomer or a homomer. After doing this, rather than just knowing it's a homomer, I wanted to see if we could see the specific number of subunits in the complex that it forms. So out of those protein homomers that I had earlier, these are broken down into different sizes. And unfortunately, once I folded these, not all of these generated high quality structures. They just mightn't be well represented within the databases, or they might have some odd folds in them. But regardless, using a cutoff of 0.6 for one of the quality metrics, I was still left with 122 homomers there. Then with these proteins here, I went and built these plots here, looking at the quality of the metrics across them. So here, for example, this one here was a trimer, and we can see that this one here peaks when 3 subunits are used using the IPTM score that I mentioned earlier. However, I also checked a range of other metrics as well for checking the quality of the interfaces, things like the P.Q score and some other metrics. And what we can see here is that each of these metrics either peaks or troughs when the correct number of subunits are used. I did a quick comparison across these 121 homomers of the different metrics that I showed there. And the best metric I found was this IPTM score, which gives us a precision of about 90% in predicting the number of subunits for our homomeric protein. So now that we have this really cool method that can be used to predict the number of subunits that a phage protein forms in a complex, I wanted to try and use this to investigate some unknown phage proteins. And one protein that I looked at was this RexB protein. So this is an unknown protein within the bacteriophage lambda, which is a model organism. And people have had some different ideas about what it might do, that it might be an anti-cell death gene, that it might prevent protein degradation, but we really don't know what it does. So using my method, what I actually found was that this particular protein here actually forms a complex with 5 different subunits. Once I modeled this protein with 5 different subunits in it, what I actually found was that it forms this really interesting protein where it has a channel through the middle of it, which I thought was quite interesting. So I wanted to go and investigate it a little bit further. And what I did next was I looked at the hydrophobicity of it. And what I noticed was that this protein here is quite hydrophobic around the center there and hydrophilic on the ends, which is consistent with what we know about membrane proteins. So what this might indicate is that this might be a really cool mechanism that the phage is using to infect its bacterial host, where it's making these like membrane channels that it's inserting inside the bacteria's membrane. This is something I would like to investigate further, and I currently have some collaborators that are testing this experimentally in their lab. But just before I wrap up, I just wanted to show you that I've generated lots of really cool protein structures using this method. And I'm calling this method the phage homomer likelihood estimated generator method, which is short for PHLEM, because we call all of our bioinformatics tools words starting with PH in my field. So in conclusion, what I've shown is that AlphaFold can help us derive phage protein complexes and help us to understand them better. Using the methods I've described, we can predict whether a phage protein oligomerizes and specifically how many different subunits are used in its complex. And in doing so, we can potentially gain better functional insights to some unknown phage proteins. I just wanted to acknowledge my lab, the Flinders Accelerator for Microbiome Exploration in Adelaide, Australia, as well as another group that I've been working with and visited last year in Germany, the Viral Ecology and Omics group, as well as a range of different groups that have funded me. And also, I'll still be around today. I've got a poster, so please come and talk to me. And I've also got a poster next week at APBJC. Yeah, so thank you.
Audience, if you have any questions, you can ask in person, and for online participants, you can write your questions in the chat box.
New microphone. Okay. Hello.
You can speak from your.
Oh, okay. Yeah, I have a question. So, how are you gonna tackle the problem with heteromeric protein complexes?
So that's something we're still working on. That's kind of without the scope of this protein here, this particular project here.
Okay.
But yeah, you're right. We could maybe do a similar approach of looking at the quality of the interfaces with heteromeric complexes. I've also seen some other interesting work lately with like Rosetta Fold Light, where they've screened for interactions between proteins using that model there. So there's lots of options to look at heteromeric ones, and yeah, that's really interesting and something we're still trying to scale up towards.
Okay. So you used only PDB structures in your project?
Yeah, so I use phage proteins I could find in the PBD just to kind of like validate the method that I'm using. But that is something that I find like quite challenging, right? Because like a structure might be uploaded to the PBD as a monomer, but in reality, like, you know, in different conditions, it could form a different type of complex. So, you know, some of the proteins there, I had to go and read the papers to see, like, you know, is it a monomer always, or is it actually something else sometimes? So I think that's one of the big limitations for this. So it's still something I'm trying to find is some good data to try and use as validation for this kind of approach.
Yeah. So are you gonna try to predict non-PDB structures, because there's many sequences that don't have a structure, right?
100%.
In PDB.
Yeah.
So you plan to approach them?
Yeah, so some of those proteins I showed on the slide previous with the really cool structures, there's some non-PDB proteins that I've taken that aren't deposited already, and I've made predictions for them. And I have some collaborators at the moment who are, you know, doing some experiments with things like native SDS gels and things like that to see if that is actually the homomeric state of those proteins, that like whether the prediction is correct for those non-PDB proteins.
Okay, thanks.
Thank you.
Okay, so we have one of the virtual participants question. So it is from Jatin Joshi. So he asked, can you explain more about the subunit steps before AlphaFold?
Yeah, so essentially, like you generate the multiple sequence alignment, and just at the top of the alignment that you generate using like collab search, there's like a spot where it specifies how many subunits are in there. And I just swap out that number for the number of subunits that I'd like to fold before I give it to AlphaFold later on.
So thank you so much.
Yeah, thank you.
So we can have the next